In this video, I'm going to go over the process of protein synthesis, and this is the instructions for the project you're going to do for this. So I'm going to go through an example of the project, explain what everything means, and then you're going to go through and do the same thing on your own. Take a picture of what you've created, and then submit it to Blackboard. Okay, so protein synthesis. Synthesis means to make, so this is what your body's going to make proteins. This is also in chapter 8 of your textbook. All right, so what is a protein? A protein is a structure made of amino acids. So think of amino acids as Legos, and there are 20 different types of amino acids. So think of 20 different colored Legos. Amino acids, when you put them together, you have a peptide here. These are peptide bonds between the amino acids. So this would be the process of putting the Legos, connecting the Legos to each other, and then based on the order of amino acids in this peptide, the protein's going to take a very unique shape. Amino acids are going to bond with each other and so on. That's beyond the extent of this class. But what you need to know is that based on the order of amino acids, the proteins then take a very unique shape, which then allows them to carry out their function. So proteins are the most diverse molecule in your body. You have thousands and thousands of different proteins. And again, the shape they have allows them to do their job. So if you change the shape, you change their function. And one way you can change their shape is if you don't have the amino acids in the correct order. So just to give you an idea of how diverse proteins are, here are just some examples. And again, this is an anatomy class, so I'm not expecting you to know all these, but just to get an idea. Your blood carries oxygen on a protein called hemoglobin. You have little channels within your brain and nerves. Every thought you have involves protein. There are enzymes. Enzymes break things down. Enzymes do several different things in your body. Those are proteins. The process we're going to talk about of protein synthesis actually involves the use of proteins. So you're not only making proteins, you're using proteins to make the proteins. Your immune system has proteins. Most of you have probably heard of antibodies. Again, cellular messengers. How do your cells talk to each other? They need proteins. Your muscles, your heart, your skeletal muscle, your digestive system that has smooth muscle, those are all made of proteins. So a very diverse and very important type of molecule in your body. Okay, so what we're going to do now is go through the process of protein synthesis. You might want to watch this video through once and then go through it again and then stop at the different parts where I give you instructions of what to do. So then you can watch it, do it, start the video over again. Okay, so the first thing you need to do is you need to find a piece of blank white paper. I guess it could be any color, but you're going to have to draw a line on it. So you're going to take an 8.5 by 11 normal size piece of paper, take that piece of paper, and then you're going to fold it in half. Okay, you're going to fold it in half. So this is kind of replaying your kindergarten years. The beginning is. So you're going to fold that piece of paper in half. And then the next step, and sometimes this gets confusing, you're going to fold each half in half. So you're going to take this half here and take this and fold it back. Okay, so in this picture I folded that half back. You're going to do the same thing with the other side. You're going to fold that back. So if you've ever made an old-fashioned paper airplane, this is how you kind of started. So again, this is where people tend to screw up. You fold this half back, fold it till it meets here, crease it, do the same thing on the other side, so you have something that looks like this. Okay, so when you would unfold it, you'll have 4 different columns. Okay, 4 different columns, each separated by a crease. Each separated by a crease. So it should look like this. Okay, so you got 4 columns. This should fold over to here. So if you need to stop the video to do that, go ahead and do that. Okay, so each of these columns is going to represent something different. So over here in this corner here on the far left column, you're going to write DNA and in parentheses put the word gene. This is going to represent the coding sequence of the gene. What I'm not going to show you here is the regulatory sequence. The regulatory sequence is the part of the gene that turns it on and regulates how active it is. Okay. Over in this far column, it's going to say DNA and it's going to say complementary base pairs. I know that's hard to read, but it's complementary base pairs. This should ring a bell for you from the last section where you actually created the double helix of DNA. Remember how the nucleotides paired up in complementary base pairs. A went with T and C went with G. All right, so you should have your paper unfolded like this. DNA gene over here, DNA complementary base pairs over here. Now, what we're going to do is we're going to create a gene, and a gene gives us the instructions for a protein. Okay? A gene gives us the instructions for a protein, and remember, a protein is made of amino acids. So the gene is going to give us the instructions for the order of the amino acids that make up a protein. So it takes three nucleotides to give us the instructions for one amino acid. Okay. One more time, it takes three nucleotides to give us the instructions for one amino acid. So what we're going to do is we're going to make a very short protein. Our protein is going to have five amino acids, okay, and that's all. So now your gene is not separated by these lines, but here, and yours should not have the same letters as mine. Yours should be unique. So if I get one and I see it's exactly what I did or very close to what I did, you're not going to get the points. I want you doing this on your own so I can check to see that you did the steps correctly. Okay, so we're going to have a gene that, because it's going to be 5 amino acids long and it takes 3 nucleotides to give us one amino acid, it's going to be 15 nucleotides long. So you're randomly going to put 3 nucleotides, then draw a line, because that tells you that's going to be the instructions for one amino acid. Then another 3, all right, and again, you can use A, T, C, and G. Randomly do it. Again, your order should not look like mine. So when I look at your picture, I want to see something unique. So use any of the 4 as random as you can to create a gene that's going to be 15 nucleotides long. Okay, so again, you may have to stop the video to do that. So again, 3 nucleotides align, 3 nucleotides align, any combination of A, T, C, and G. And again, it should not look like mine. So that'll be the first step. Now, just to practice, fold your paper back up, and on that one column, that far left column, again where it says DNA complementary base pairs, I want you to do the complementary strand of that DNA. Okay, so notice the A, here's a T. So just like your model, you're going to mimic that model. So take whatever your nucleotides are and do the complementary base pair strand of the DNA. So here's the DNA. It's sitting in the nucleus. Okay. The DNA, or your genes, are the instructions for the proteins. So they're basically the blueprints of your body. Okay. So we've got this strand of DNA. Imagine this is a double helix. You kind of got to use your imagination. You got this double helix strand of DNA in the nucleus. And think about it. If you had blueprints to a very important product that you were going to make, you don't want that original blueprint to leave the nucleus, or to leave your safe in your very important office, secured office in the middle of your factory. So what the body's going to do, the body thinks the same way. We don't want the original to leave. We're going to make a copy. All right, so when that regulatory sequence says, it's time, gene, we need to make some of that protein, then what's going to happen is the DNA strand is going to unwind. Now you're going to represent that by doing that. Okay, so you're going to unwind your DNA double helix. Okay. Now what you've done is you've exposed the coding sequence of the DNA. So what's going to happen now is we're going to use a new molecule called RNA, ribonucleic acid. There are several forms of RNA. The one that we're going to deal with is called messenger RNA. So here's your top left corner. There's your crease. On the other side of that crease, but right along the crease, we have messenger RNA. The only thing I care about that you know about RNA is that there are no T's in RNA. So wherever you would put a T, you're now going to put a U, and that U stands for uracil. So there's a different base. There's no thymine. Wherever you would put thymine, you're going to put a U for uracil. Okay. Your book mentions the enzyme that's going to do this, which again in itself is a protein, is called RNA polymerase. And what it's going to do, there are these nucleotides, these mRNA nucleotides in your nucleus. They're going to start to pair up. They're going to start to form a complementary base pair strand of mRNA with the gene. Okay, so for my example, and again yours should not look the same, here's what happens. The mRNA nucleotide, again it's an A, which we're used to saying a T would go there, but since we're talking about mRNA, it's a U. Go down and form your mRNA strand, your messenger RNA strand. Again, complementary base pairs. And make sure you do it correctly. Wherever there's an A now, though, you're not going to put a U, or you are going to put a U. You're not going to put a T. Now, where you have a T, you are going to put an A. Adenine still does exist in mRNA. So take time here and go through and make your mRNA strand. We are making a copy, so think of it as a copy machine. The DNA gene is your original, and you're making a copy out of mRNA. Okay, go to the bottom here. Okay, this is just for your notes to help you study or review for this part of the test, or have what you need in order to review. Go to the bottom here. You should have left some room here. If you didn't, just write it on something else. But this process of going from DNA to mRNA is called transcription, and it happens in the nucleus. So you may see me use this representation on the test, ask you what it is. It's transcription. Where does it happen? It happens in the nucleus. Okay. And again, that should go on the bottom here. Or you can put it just on the other side of the piece of paper, just so you have something so you can see it if you went down too far with your gene. Okay, so what's the next step? The next step is we have to get that copy out to the cell to actually produce the protein. Just like if you had a factory, you have to take a copy of those blueprints and send it out to the factory to be made. So what you're going to do here, this strand of mRNA is going to leave the nucleus and go to the ribosomes. Remember, ribosomes, their function, protein synthesis. So what I want you to do is just draw arrows here. It's the same strand. Okay, it's the same strand. It just represents this strand moving out of the nucleus to the ribosome. And you can actually add a note there if you want to write nucleus to the ribosome. That may help you. So this is the same strand. You're just going to move it over to the edge of that column right on the crease. But notice it is the same exact strand. Okay, it is the same exact strand. Okay, now what's going to happen? Now what's going to happen is, and where I am on this drawing, just so you know, is right here. I'm showing you what's going to happen with this first, instructions for the first amino acid. All right, those first three, every three nucleotides and on the mRNA, the instructions for one amino acid is called a codon. Okay, it's called a codon. So UAG, that's the instructions for one amino acid, that's a codon. The next three, another codon. Okay. The next molecule we're going to use is another form of RNA, and that is called transfer RNA. That's a T. Okay. Transfer RNA, same principle applies. You don't have T's, you have U's. But it's unique in the sense that each molecule of tRNA is made of actually 3 nucleotides. Okay, on one end, and on the other end is one of the 20 amino acids. The amino acid that is located here is dependent on what these 3 letters are. These 3 nucleotides are called the anticodon. Now, how do I know what amino acid is going to go right here? What you need to do is look at your codon. Okay, look at your codon. So in this case, it's UAG. Then go to page 176 in your book and look up, find the codon. Okay, find the codon. So what we had, UAG. Now, I picked a bad one, but this one's called a stop codon. If you get a stop codon, there's another one here. Just go to the amino acid that's in the same box, this box right here. So in this case, it is tyrosine. Okay, so for UAG, it is tyrosine. that is represented by a 3-letter abbreviation. That is one of the 20 amino acids. Okay, that is one of the 20 amino acids. So you find your codon, UAG. It's going to tell you which amino acid is attached to that transfer RNA. So you find that codon, okay, UAG. That means the tRNA that is AUC is going to fit there, and the amino acid that's attached to that tRNA is tyrosine. So you look up the codon, draw in your tRNA. Based on this chart, you know that the amino acid that it goes with that codon is tyrosine. Now here, notice I made tyrosine a rectangle. You have to come up with a unique shape for each different amino acid. Their shape is important. Okay, their shape is important. So what you're going to do is you're going to do the same thing for all 5 amino acids. Find the codon on this sheet, on this chart, use the 3-letter abbreviation, and then come up with a shape for the amino acid that goes with that codon. Oops, I put that in again. Okay, so here's what you end up with. Here's the one we just did. You look up for this one, GCU, for me. It should not be the same for you. Go back to that table. Okay, what amino acid goes with that codon? In this case, it's alanine. Alanine's a different amino acid, so it's going to have a different shape. Do the same here, here, and here. So you should have 5 amino acids. Now, if just by chance I got tyrosine again, let's say down here, I have to make tyrosine the same shape here as I did here. Now, I want you to notice there's a bond between these amino acids, and that bond is called a peptide bond. Okay, that bond is called a peptide bond. What we have now done is we have made a peptide, we have made a protein. Okay, we have made a protein. Now, what's going to happen to that protein? This kind of be review. That protein is then going to go to the endoplasmic reticulum, and it's going to be shaped. And the shape of that protein is going to be based on the relationship or the order of these amino acids. They bond with each other. They bond in several different ways beyond what you need to know in this class. You get a very unique shape because of the order of those amino acids, and then you get a unique function because of that shape. Okay, because of that shape. Okay, so to review what we just did now, on the bottom here, okay, on the bottom here, you're going to write this, or on the back so you remember. The process of going from messenger RNA to tRNA to protein is called translation, and it occurs at the ribosomes. So again, this process here of going from mRNA to tRNA to the protein, or you could skip a step there, going from mRNA to the protein, is called translation, and it happens at the ribosome. Okay, so if you haven't, go through that process of doing that. And this again is just showing you that this whole structure here is a protein, and they are bonded together by peptide bonds. And like I said, ignore this. This is just your this is the 4th column. That was just part, remember, the complementary base pair of your gene. So just ignore that for now. Remember, this protein will go to the endoplasmic reticulum, be shaped, go to the Golgi apparatus, be shipped to where it needs to go to do its function. Okay, to do its function. Okay, so now we're going to talk about, after you've done that, do all of that first, we're going to talk about what a mutation is. Some mutations you're born with, other mutations you accumulate over time. When we get into talking about cancer, we'll talk about things called carcinogens. Carcinogens you hear about can cause cancer, but they do that by causing mutations or changes in the order or the structure of the gene, which can cause changes in the protein. So what you need to do, let me show you the process. Don't do it until you're sure what you're doing. So what I'm going to do, you're going to pick, and you can do the same thing. You can pick the 5th nucleotide. So 1, 2, 3, 4, 5. I'm going to change that. I'm going to mutate it, and I'm going to change it, and you don't have to change it to a T. You have to change it to a different letter, though. If it's a G, it can be A, T, or C. It can't be the same letter. So you're going to change that. Okay, you're going to change that. We have now mutated the gene. In the case of skin cancer, that could happen from sunlight. That would mutate a gene. Okay. Smoking mutates genes. People that have a family history of cancer, they may be born with a mutation in a gene. And we'll get into those genes and discussing those later. Next, what you're going to do, which is what I did here, you're going to take that mutation. If we have a mutation in the gene, then you're going to go ahead and say, okay, what does that do to my mRNA? Well, now I have a T here, so that T is going to change to an A. So that will move over. You change this to whatever letter this is. Now you have to look up the new codon. The new codon here, it's no longer GCU, it's GAU. Okay, GAU. That codon is going to change, in this case, what tRNA binds here and what amino acid is here. So it's no longer alanine, it's asparagine. And that has a different shape, which I drew here. Okay. So now what's happened is, and I didn't draw this in because I didn't want to add too much to the drawing, you now have a protein, that has a different order of amino acids. And what that means is, when it goes to the endoplasmic reticulum, it's going to be shaped incorrectly because the order of amino acids has changed, which means its function's going to change, and it's not going to do its job correctly. We'll talk about cystic fibrosis. There's these little things called chloride channels that sit in your cell membrane. They're chloride channels. They have mutation in a gene. The order of the amino acids of the protein of the chloride channel has changed, and now that chloride channel doesn't work. So you can, many times with a mutation, you lose the function of that protein. Now in some cases there is what's called a silent mutation. A silent mutation is when you happen to change an amino acid, or sorry, when you change a nucleotide. When you change a nucleotide, but it just so happens that it doesn't change the amino acid. So you have a mutation in that you have a change in the order of nucleotides, but by chance it doesn't change what amino acid you look at. Okay. And what happens, let me go back. If you look at this closely, you'll notice, like phenylalanine, there's more than one possible codon that gives you phenylalanine. And here, leucine, there's actually 4 different codons that give you leucine. So if you get a mutation where you go from CUU to CUC for the codon, it doesn't change the amino acid. Okay? It doesn't change the amino acid, so it's not going to change the amino acid, it's not going to change the protein. That is a silent mutation. Okay, that's kind of the end, but I want to explain what you're going to do. What you are going to do is you're going to go through this on your own, okay? Fold your piece of paper up, go through the steps just like I did in this video. Again, you need to randomly pick your order of your gene. It needs to be 15 nucleotides for 5 amino acids, but it should not be the same letters I used. And then you're going to go through the process of transcription over here to translation to the point that you end up with a protein, and I should be able to see that protein. Then you're going to do a mutation, and you're going to change, like I said, the 5th nucleotide and see what happens. See what happens to your protein. Is it a silent mutation, or is it a mutation that's going to show itself in terms of changing your protein structure? As we get into cancer and we get into other diseases and we look deeper into genetics, you just have to have a basic handle on this process and understanding what happens with a mutation. Okay, so what you're going to hand in is you're going to take a picture, and please, please, please make sure the picture takes up the whole frame if you're taking it with your phone, and that it is in focus. I have to be able to read all of this. So you can take the picture, You can put it into a Word document. Okay. I'll give you a Word document to put it into. Make sure it takes up as much of the page as possible and make sure I can read everything. If you can't read it on your Word document, then there's no way I can read it. Okay, there's no way I can read it when I'm trying to grade it. All right, so I'm looking for something that's big enough that I can read, take up the full piece of paper, and it has to be in focus. Okay, it has to be in focus. So make sure it's very clear. If it's not clear, I'm just going to send it back to you. All right, if you have any questions on this, let me know. I'll put up a discussion board, so if people have basic questions, they can ask. You can ask, either classmates will respond to you or I will respond to you with an answer. All right. Again, you may have to go through this more than once. Have a great day.